A-level items for 2026 samples. As we go through, take note of the disruptions in the process. A tomato production program in Masaka district was established to improve household incomes and supply fresh fruits to urban markets. During routine field inspections, agricultural officers discovered that some farmers had applied a herbicide intended to control weeds. Further laboratory investigations revealed that the herbicide affected the activity of NADP plus reductase in chloroplasts. The district also experienced prolonged drought during the fruit development stage. Researchers found that drought conditions affected the production of abscisic acid in leaves. In addition, some farms were exposed to smoke from nearby charcoal burning sites. Investigations showed that the smoke affected ethylene production in tomato plants. As the season progressed, fruit production declined and marketable yields reduced. To minimize losses, farmers harvested fruits immediately after the first signs of ripening. Farmers sorted and stored mature fruits separately from unripe fruits. Farmers irrigated their crops. Analyze the data table. Compare the normal and affected. Look out for the changes in the parameters. Task Part A. Analyze how NADPH production, RuBP concentration, and carbon fixation rate contributed to reduced tomato yields. Part B. Evaluate the effectiveness of the farmer's interventions in reducing post-harvest losses and maintaining tomato productivity. Analysis. As usual, we are going to find out what happened, why it happened, and how. Reduced NADPH production. NADPH production decreased from 100 to 42. This occurred because the herbicide inhibited NADP plus reductase, reducing the conversion of NADP plus to NADPH during the light-dependent stage of photosynthesis. Reduced NADPH limited the reduction of glycerate 3-phosphate to triose phosphate in the Calvin cycle, decreasing carbohydrate synthesis. Consequently, less glucose was available for respiration, fruit growth, and development, leading to reduced tomato yields. As we continue, get the book. Self-study, A-Level Biology today, if you want to succeed. Reduced RuBP concentration. RuBP concentration decreased from 94 to 51%. This occurred because prolonged drought increased the production of ABA. ABA stimulated the movement of potassium ions out of guard cells, causing water to leave the guard cells by osmosis. The guard cells lost turgidity and the stomata closed. Stomatal closure reduced carbon dioxide entry into the leaf, lowering the rate of RuBP regeneration in the Calvin cycle. Consequently, fewer carbon dioxide molecules combined with RuBP, reducing photosynthesis and fruit production. Reduced carbon fixation rate. Carbon fixation rate decreased from 96 to 46%. Reduced RuBP concentration and limited carbon dioxide availability lowered the formation of RuBP carbon dioxide complexes by Rubiscio, reducing the incorporation of carbon dioxide into organic compounds. Consequently, less glucose and other assimilates were produced for fruit growth and development. In addition, excessive ethylene stimulated premature fruit ripening, shortening the period available for fruit enlargement and accumulation of food reserves. Therefore, fewer fruits reached market maturity, 39 compared to 88%, resulting in reduced yields. Evaluation. Harvesting fruits immediately after the first signs of ripening. Early harvesting removes fruits from the field before prolonged exposure to excessive ethylene can accelerate over-ripening and spoilage. This reduces post-harvest losses and increases the proportion of fruits reaching consumers in acceptable condition. Therefore, the intervention is effective in maintaining marketable yield. However, harvested fruits continue producing ethylene, which can still accelerate ripening during storage and transportation, leading to deterioration and reduced shelf life. Recommendation: cold storage. Cold storage lowers the rate of respiration and ethylene activity in fruits. This slows ripening and deterioration, extending shelf life and enhancing the effectiveness of early harvesting in reducing post-harvest losses. Sorting and storing mature fruits separately from unripe fruits. Ripening fruits release ethylene gas, which stimulates ripening in nearby fruits. Separating mature fruits from unripe fruits reduces ethylene exposure and delays ripening of the unripe fruits. Consequently, spoilage is reduced and more fruits remain marketable for longer periods. However, ethylene may continue accumulating within storage facilities from the mature fruits, causing continued ripening and eventual deterioration of stored produce. Recommendation: use of ventilated storage facilities. Ventilated storage facilities continuously remove accumulated ethylene gas from the storage environment. This lowers ethylene concentration around stored fruits, slowing ripening and enhancing the effectiveness of fruit separation. Irrigating crops. Irrigation supplies water to plants and reduces drought stress. Adequate water lowers ABA production, promotes stomatal opening, and increases carbon dioxide uptake for photosynthesis. Consequently, photosynthetic activity, fruit growth, and productivity improve, making irrigation effective in maintaining tomato production during dry periods. However, excessive irrigation may cause waterlogging of the soil. Waterlogging reduces the amount of oxygen available to root cells, lowering aerobic respiration and ATP production. Reduced ATP limits active uptake of mineral ions and water by roots, leading to poor plant growth and reduced productivity. Recommendation: installation of drainage channels. Drainage channels remove excess water from the soil and prevent waterlogging. This maintains adequate soil aeration, ensuring sufficient oxygen supply for root respiration and efficient absorption of mineral salts and water. As a result, the effectiveness of irrigation is enhanced while minimizing its negative effects.